We're going to get started. So hello, everyone. Welcome. Thank you for joining us this afternoon. My name is Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. And I'm joined today by a number of team members from Satellos Biosciences to talk about their investigational product, SAT3247. So they're going to talk a little bit about their investigational product and then some of the biology that helps kind of guide this approach, which, it's novel. We think it's helpful to better educate the community kind of around the rationale for how they're developing a potential therapy for treatment of Duchenne. So with me today on this call from Satellos, we have Ryan Mitchell, who's the SVP of Scientific and Medical Affairs. We have Jordan Dubow, who is the Chief Medical Officer. We have Michael Rudnicki, who's the Chief Discovery Officer and co-founder of Satellos, and Frank Gleason, who is their CEO and co-founder. So we are recording this webinar. So if you have to drop off or if you have family members who you think this would be relevant to, we should have this posted up on PPMD's website within the next week. So we encourage you to share that as needed. And at the end of the talk today, we are going to have an open Q&A. So as the team from Satellos is going through the presentation, if you have questions about what they've presented or some of their work, please feel free to submit that in that chat function at the bottom of your screen. We'll try to get through as many questions as we can in the Q&A. And if we run out of time or are unable to address your question, please feel free to send it to the team here at PPMD and we'll forward it along to the group at Satellos. But with that, I think we can dive in. And so I'm going to hand it over to Frank to get us started.
Well, thank you, Eric. And thank you very much to PPMD. We're absolutely delighted to be here today to speak to parents and family members and patients about our program and our new small molecule drug. And we're really grateful to PPMD for years of support in helping us to get to where we are today. So as Eric said, there are a number of members of the Satellos team on this call today to both present information to you and then to answer your questions. I'm really pleased that Dr. Rudnicki, our co-founder and a very long-term colleague of mine, is able to join us. Dr. Rudnicki is really the discoverer of our technology and our approach to treating muscular dystrophy. And between the two of us, this has been a journey now that is coming on its 7th anniversary early in the new year. And by that point, we will be able to begin treating patients in a clinical trial. So we're so delighted to be this close and to have this opportunity to speak to you. So also on the call, as Eric said, is our Chief Medical Officer, Dr. Jordan Dubow. Dr. Dubow has a really lengthy background in the neurosciences and neuromuscular area and has been involved in multiple programs, including in Duchenne, that have led to drugs being approved by the FDA. We have Courtney Wells on the call, who is our head of clinical development and clinical operations. And Courtney has been with us a little over a year and largely responsible for helping us get so quickly into clinical development. And Ryan Mitchell, who is our head of medical and scientific affairs. And Dr. Mitchell was the first employee to join Michael and myself. He is a stem cell scientist who has been really instrumental in helping us communicate what we're doing to a broader audience. And Ryan will be leading the presentation today. So I'm going to turn it over to Ryan. And I just am so pleased that there are so many of you on the call today. And we hope you'll find what we're talking about of interest to you. And we're looking forward to this interaction. So over to you, Ryan.
Thanks, Frank. And I'll echo all of Frank's thanks to PPMD and of course everybody joining us today, and equally excited to share the Satellos story with all of you. So we'll start off with our prerequisite disclaimer slide. Not the most exciting one for today, I promise you. So Satellos, Frank and Michael and the team that they've developed really started this project with a question, and we think a unique question. And that was, what should a novel medicine for Duchenne truly look like? And we had a number of criteria that we used to answer this question. To us, a novel treatment should be safe and highly tolerable. It should also be easy to administer. It should not be limited by your genetic mutation. We also believe it should not be limited by your ambulatory status. Importantly, it should prevent further muscle loss, but perhaps even more importantly, it should have the potential to replace muscle that has been lost due to Duchenne. And we're very excited to say that SAT3247 is being investigated to determine its potential to meet each of these criteria. Already, we believe that we have a potentially efficacious dose based on animal models that's expected to be below toxicity levels. 3247 is being developed as a small tablet for oral administration, a pill, and a small pill at that. We do believe that there is potential to benefit individuals with all of the different mutations that we see in Duchenne. We also think that there's potential to benefit individuals at different stages of the disease process, and of course, different stages of ambulatory status. And excitingly, 3247 has been specifically designed to repair muscle damage when it happens. And maybe even more excitingly, it's been designed to potentially regenerate damaged or even lost muscle. Now, all of this thinking, as Frank alluded to in his introduction, really stems from one person on our team, and I think together we've helped develop it. But these were really questions and ideas that were posed by Dr. Rudnicki. Dr. Rudnicki thinks very differently about Duchenne, and he does that because he's a stem cell biologist. He has studied how different tissues in the body are able to regenerate and repair in response to damage. And in thinking about Duchenne and understanding that Duchenne is certainly a disease of muscle damage, he couldn't really calculate how this disease made sense. And so he had some seminal thoughts and questions that he thought of. And the first is that Duchenne is dystrophin, is missing from birth. However, why does it take years to see symptoms? We know the disease gene, we know the mutation, and that mutation is there from day one. And yet symptoms take years to present. And why do muscles function in the beginning, but not in the end, if everything's related to dystrophin in the muscle fiber and dystrophin's missing right from day one? Why is it that muscle without dystrophin in early young children with Duchenne is still primarily functional, but later on in life, it's not? And why is the muscle tissue progressively lost on a time scale of years? Dystrophin is missing from day one, but it takes years and years for the muscle tissue to be lost. It's not immediate. And all of these sort of questions and ideas and thinking that Michael had really led him to consider that dystrophin must have another job, a different role from the one that I think we all understand in the muscle fiber that's not getting done. So what do we know about dystrophin? And what have we been thinking about as a community for quite some time? Well, we know that dystrophin has a role in the muscle fiber. And I'm sure you've all seen this diagram on the left-hand side of the slide many, many times. And we know that dystrophin functions as some kind of a shock absorber or a structural component of muscle fibers that helps to keep muscle tissue intact during use or during muscle contractions. And what we know in Duchenne is that the loss of dystrophin in the muscle fiber leaves those fibers vulnerable to damage. We know that regular daily activity can lead to damage in muscle fibers, tears and rips in the muscle tissue. What that doesn't explain, though, is why the muscle doesn't simply repair the damage as it happens. Muscle is a highly regenerative organ. When we damage the muscle tissue, we can repair it quite well. But in Duchenne, we know that this is not happening. What Michael's team discovered is that dystrophin has another role. This role is entirely outside of the job that dystrophin takes care of in muscle fibers. And this unique role or job is actually related to how the muscle tissue responds to damage. And it turns out that dystrophin provides a signal that helps to control how the muscle repairs and regenerates. And it does that by providing a signal in what we call muscle stem cells. Now, muscle stem cells live inside the muscle tissue, and they have one job in life. That is to respond to the demands of growth or damage to make new muscle tissue. That is all they do. Otherwise, they sit around in the muscle tissue and sleep. But when the muscle tissue is damaged, they wake up and they have to go through the process of cellular division to make more cells. And they do that in a very special way. And we as stem cell biologists call that a stem cell asymmetric division. And really what that is a fancy term to describe one stem cell dividing in a similar way to this picture on the left is showing you. It divides to produce two different cells. It makes one copy of itself, which we've colored in green on the right-hand side of that figure. And then it makes a more specialized cell, and we've colored that in purple. And we call that specialized cell a muscle progenitor. Now, those progenitor cells are really, really important for muscle repair and regeneration because they can go on to divide many, many, many times and make lots and lots of new cells. And it's those new muscle cells that go on to repair damaged tissue. And so now you can imagine dystrophin is playing a pretty important role for the repair and regeneration of muscle. It is part of the signaling that helps tell the stem cell, hey, we need new tissue, we need new muscle cells, you need to go through one of those asymmetric divisions and make a progenitor, and then that progenitor can go to work. So this is how muscle normally works when dystrophin is present. However, when dystrophin is missing, now that signal is lost. So it's no longer playing its role or doing its job in the muscle stem cell. And in the absence of that signal, now the cells behave differently when they get called to action by damage. Instead of going through that asymmetric division process, now the cell gets a little bit confused. It instead makes copies of itself. So no longer does it produce one progenitor and one stem cell. Instead, it divides symmetrically and makes two identical copies of itself more often than not. When this happens, we have an impairment or a loss in the production of those extremely helpful and useful progenitors. And this leads to ineffective or less effective muscle repair and regeneration. Over time, what this means is that the muscle cannot properly repair itself. And that muscle tissue, after being damaged over and over again, is progressively lost. And so if we take all of these ideas and think about it in sort of a logical framework, these two key roles for dystrophin, what we see is that dystrophin loss leads to damage and impaired or less effective muscle regeneration. So the lack of structural dystrophin or shock absorber dystrophin, as we've heard for so long, in muscle fibers, that's what leads to the damage occurring in the first place. But it's the lack of signaling dystrophin in the stem cell that leads to the reduced ability for the muscle to respond to that damage and repair itself and regenerate that tissue instead of losing it. And so it's this inefficient regeneration response that we believe leads to the progressive loss year over year of muscle tissue. So we ask the question, what if the regeneration system could be restored to keep up with the damage that is occurring? And that is why we designed SAT3247. So we created this molecule with the intention to try and restore the regeneration process so that Duchenne muscle can keep up with the damage. 3247 was discovered in laboratory experiments that we did in Michael's lab as a way to replace that missing signal from dystrophin. 3247 inhibits a protein called AAK1. AAK1 is a protein in the Notch pathway. And when you inhibit AAK1 or start to shut it down with 3247, that replaces a missing signal that can achieve that asymmetric stem cell division, just like dystrophin was. So it's not as if 3247 looks like dystrophin or is a piece of dystrophin. This is an entirely independent way just to replace the lost signal, to tell the stem cells, hey, we need you to divide asymmetrically so that you can produce a progenitor cell, and that progenitor cell can actually go and do its job and repair and regenerate that muscle tissue. That's all 3247 does. It helps to instruct the stem cells to divide properly and go through the regeneration process. So lots of information on how 3247 works and why one might want to restore regeneration. Now I'm going to show you some preclinical data that demonstrates what happens when you do restore regeneration. We've released a great deal of data on our website and through presentations in the MDX mouse, but today I'm going to focus on some work in a DMD canine model. So we were able to perform a study in 2 Duchenne canine models. This was a 4-month study in 9-month-old dogs, and these dogs were given an oral dose of 3247 for 4 months. We measured a whole bunch of things in this study, but most important for today, we did look at the effect on the muscle tissue itself through histology. And of course, we looked at the effect on muscle function through strength testing. So that's what I'm going to focus on today. So we'll start with histology. So to orient you to these images, on the left, we have a piece of biopsy material from one of the animals prior to treating them with 3247. That's on the far left. The next image in is a piece of muscle tissue from the same muscle from the same dog 4 months after treatment. And on the far right, we have a piece of tissue from a dog that was not in the study, which was a healthy dog that did not have Duchenne. What we're trying to exemplify in these pictures is overall the way the muscle looks. You can see that from some of the green staining, which is a protein called laminin. I'm sure many of you have seen staining like this before. That laminin protein really wraps around the outsides of the muscle fibers. What we're also staining for in red is IgG. And IgG is an immune molecule that really should not be present inside of muscle fibers unless those muscle fibers are damaged. So here we use IgG as a surrogate marker for damaged muscle fibers. And hopefully what you can appreciate from these images is that in our dog before treatment with 3247, there was an appreciable amount of muscle damage that we could observe. However, after 4 months of treatment, there's far less damage that we were able to see in these muscles. And I can tell you that this is from a bicep muscle, and we've now confirmed the same phenomenon in multiple other limb muscles, including the diaphragm. And so it would appear to us that providing 3247 and restoring the regenerative capacity of skeletal muscle in these dogs was able to improve the overall architecture or the structure of the muscle tissue and reduce the number of actively dead or dying fibers and or having regenerated new fibers. So the muscle overall looks much better, but importantly, we also measured the function of the muscles in these dogs. So before I walk through this data, I just want to make it very clear that the Satellos data from our two animals is presented in the green and the purple bars. And in both of these graphs, what we've done is we've taken information that was published by others in the literature, and we've overlaid our data with theirs so that we can give a sense of how our treatment with 3247 compares to the data from these other studies. The first study that I'll go through is on the left, and this was a study looking at the strength of a particular muscle in the canine Duchenne model over the months of growth and time that passed in these dogs. So this is looking from the ages of 3 months to 12 months in Duchenne muscular dystrophy dogs, as well as healthy dogs. So the healthy dogs show a strength profile over time in the gray line, and the Duchenne muscular dystrophy dogs show their strength profile over time in the black line. And then the two Satellos dogs here are in the green and the purple line. And hopefully what you can appreciate is that Duchenne muscular dystrophy dogs have lower strength than regular wild type dogs. So the black line is lower than the gray line. However, the Satellos treated dogs, after being treated, well, in fact, they started off even lower in strength than typical Duchenne muscular dystrophy dogs. And after 4 months of treatment, they were able to improve very close to the levels of wild type dogs. And so from this information, we believe that 3247 had a positive impact on the functional outcome in these animals in this study. And that positive impact is very clear when we contextualize this with the natural history of the Duchenne dog model. The graph on the right now is a little bit of a different comparison. So here we're no longer looking at natural history. Here we're looking at the comparison of the Satellos treatment versus the treatment with a microdystrophin vector. And instead of looking at age along the bottom of the graph, here we're looking at the days after these two different treatments were delivered. And the reason why we've done that is because the dogs in our study were actually of a different age than the dogs in this microdystrophin study. Our dogs, as you may recall, were 9 months of age. The dogs from this particular microdystrophin study were 3 months of age. And so instead now we simply compare the effect after treatment with the two different approaches. The data from the microdystrophin study is presented in the black and gray lines. The black line are animals, are Duchenne canine models that did not receive any therapy. And the gray line are the animals that received the microdystrophin therapy. And so we can see an improvement in strength from those animals that were treated with the microdystrophin therapy. And then you can see the Satellos animals, the green and the purple line, which are plotted on top, clearly showing that these animals were in fact not as strong as the dogs from the other study, likely because they were older and potentially much more progressed. However, after 4 months of treatment with 3247, we saw very, very comparable improvement in the strength of these much older animals, simply by restoring their capacity to repair and regenerate. So when we think about these two pieces of information together, we feel very strongly that this idea of enhancing or restoring the capacity for muscle to repair and regenerate is potentially something that could be efficacious across all streams of Duchenne. Now, where are we now? So at this time, I'd like to invite Dr. Jordan Dubow to come and make some comments and walk you through where we are now in our clinical platform.
Thanks, Ryan. I think it's amazing work that the preclinical team has done, and we're so excited to actually be in the clinic. And as Frank alluded to, a lot of that credit belongs to Courtney Wells, who's on this call, who's done a Herculean effort to get the drug into the clinic as quickly as possible. And so, right now we currently are treating kind of our first in human phase 1 study, and that's being done in Australia. And those who know a little bit about drug development, there's kind of different reasons why you can potentially go to other countries first before the US, but we're right now in Australia. We've dosed a number of cohorts already of healthy volunteers first, where we're looking at kind of blood levels of the drug, testing the safety of the drug. We first start with one dose, going higher and higher and higher over time. And then we move to multiple doses where these healthy volunteers will get multiple doses. So far, we like what we're seeing so far. And after that, as part of this study, we actually are going to dose a cohort of adults with Duchenne. And so that, as you can see, will happen later this year. And we're hoping to also look at kind of blood levels of our drug in patients, adult patients, as well as look at some potential biomarkers or see kind of early signs of, could this drug have some benefit? After that, we're really quickly looking to move kind of into multiple studies here in the United States, as well as other parts. And so, as many of you may know, there's a long kind of a lot of data, toxicology data, looking at kind of testing the safety of drugs in different species of animals, and the process of kind of putting all that data together, data from this first study in Australia, and then filing all this information to the US, to our FDA, to first start a study in older boys, and then quickly after that kind of have another filing to go younger and younger through potentially different age groups of Duchenne patients. Some of the studies will be kind of open label, and some of them will actually have a control placebo arm. As Frank said, our goal is to actually see if this drug can work in all stages of the disease, multiple ages, adults and children, both ambulatory and non-ambulatory. And we hope to be able to test that again starting in 2025. And the main goals of those kind of what we'll call them phase 2 studies is to look at different doses to see what may be the best dose, to look at certain biomarkers, and look for first signs of kind of efficacy. Are we improving motor strength, muscle strength? Are we improving function? And so we'll continue to work with PPMD to make sure that we're very vocal about when we're enrolling. We're obviously going to need volunteers, participants to go into these studies, because obviously we want to get there as quickly as possible and hopefully generate really exciting data to continue to move the program forward.
Thanks, Jordan. So I think we'll quickly flash the team here. Come off camera. So these are some of the many faces, and we're a growing team at Satellos, and we show this just to let you know that there are a large number of people on this team now that are trying to push this idea of Michael and Frank's into reality. And I'd also like to thank a lot of the groups that got behind us in early days, including PPMD, who made a very important investment in our company early on, and we wouldn't be here without them, and the growing list of partners that we continue to work with. And with that, I would like to invite Eric to come and moderate the question period.
Thank you all. That was a really great presentation. Can we maybe go back to slide 5? I think it might be easier for some of the questions I think that came in as we kind of talk through some of these pieces. So we had some questions submitted before, and then as the talk was going on, so we'll try to get to as many of those, and as they're scrolling in, I'm hoping I won't miss them as they come by. But we did have questions, and I think you already talked about this, but I think it's always good to help reiterate, because we had these questions come in both before and while we were going through this. But the expectation for SAT 3247 is that it is not limited to whether you are ambulatory or non-ambulatory, correct?
Yeah, so the goal in clinic, and then, I don't know if you want to speak to the preclinical. In the clinic, yes, we are planning on testing it, first in adults. As we know, sometimes with regulatory agencies, we have to start in adults before we move to kids. But nevertheless, we do have goals to test it in all populations. I think we have reason to believe this drug could be effective in all populations. So that, currently is our goal, pending, kind of those ultimate discussions with FDA.
And then we had a question that someone posed, I think thinking a little bit more about the non-ambulatory population, that as some of the patients age and there's some of that fatty fiber replacement of the muscle tissue, would that impact kind of the effectiveness or how the muscle regeneration could potentially work with a drug like this in those older kind of patients?
Yeah, so I think building off what Jordan had said, of course, it remains to be seen. That's something that we will have to evaluate clinically. Speaking to the science, the way that we think about this is if there's muscle, and this is really what the research has shown, if there's muscle left that can be biopsied, there seems to be an abundance of stem cells in that muscle tissue. And that, in our view, is because the muscle is still trying to respond to the damage, but the cells aren't dividing the right way. And that's really the signal for us to believe that the drug could have potential, because that type of study has been shown in non-ambulatory patients in the muscles that are still remaining, that phenomenon is there and present. And so it's why we would like to test that, and we believe there could be applicability.
And that might be a good jumping off point, because there was a question that someone asked that they had heard that some of the challenge with Duchenne was in that kind of constant cycle of breakdown and repair of muscle, that there was maybe hyperstimulation and in some of the stem cell pool being depleted. So if you could maybe talk a little bit to that kind of idea.
Yeah, so as far as we can tell from literature actually looking at patient samples, I think there's been some interesting findings historically in the Petri dish as to whether there could be reason to believe that stem cell depletion might be a reason why muscle progressively is lost. However, from patient level histology, there is no evidence to support depletion of stem cells. In fact, it's quite the opposite, which really is the entire premise of what we're trying to do.
Great. Could we maybe go to slide 10? So I think you did a good, so again, it's a newer kind of approach, and I think discussion around kind of the role of dystrophin and some of kind of the potential biology here. And so it's always a little bit challenging. I think you did a good job of breaking some of this down, and I think having Dr. Rudnicki here is helpful as well. Someone was asking about just trying to better understand how we can know that is the role, that one of the roles that dystrophin is playing. And so I think, can maybe speak to a little bit of the research that you've done to kind of get to that point. I think you did a good job of explaining the role and how that is going to work, but that might be helpful for some of the families.
Sure. Michael, do you want to comment on how your lab discovered this new role for dystrophin?
Yeah, so it was serendipity. It was an observation we made in MDX mice that lack dystrophin. We found that their baseline levels of asymmetric stem cell division, my apologies for the announcement, that the asymmetric division on cultured myofibers was extremely low compared to normal mice. We then established that this was a deficit that was due to dystrophin itself. We identified the signaling kinases that were needed to be present that bound dystrophin, and what happens to polarity in that context. And there's now a wealth of information that polarity is disrupted in the absence of dystrophin, that produces this intrinsic deficit in stem cell function. They prefer to divide and make more stem cells, so you have an overgrowth of stem cells, rather than give rise to progenitors that can divide multiple times to repair the tissue. And so this has really profound implications, and really means that DMD is also a disease of stem cells. And by modulating the stem cells with our drug, we can restore those regeneration processes to levels where we really believe will have an impact. So this is very exciting. So the notion that stem cells are exhausted or depleted is wrong. It's incorrect and is outdated, and has to be updated with this new insights.
Great, thank you.
Michael.
And then on the next slide, and Ryan, you touched on this in the presentation, but I always want to make sure that when we're talking with families and we're hearing the term stem cells and some of that, that just the separation of this is not a cell-based therapy you are delivering. And maybe you could kind of just touch on that again here.
That's great. No, that's a very good point, Eric. So we are, we are a stem cell company. We do lots of research on stem cells, but 3247 is absolutely not a cell product. This is an orally available small molecule drug, so a pill, right? A tablet that will be taken out of a pill bottle that once in the body and in the bloodstream makes its way to the muscle tissue and influences the muscle stem cells that are already there. So the drug goes into the body and then influences how the muscle stem cells behave. And maybe just a quick recap. So muscle stem cells have this one job, and that is to respond to muscle damage. And if you don't have Duchenne, you don't have any other kind of muscular dystrophy, anytime you have a muscle injury that's fairly serious, muscle stem cells wake up and they start dividing. And they have a job to do, and that is to make more muscle cells. And that's the job they're failing to do in Duchenne because dystrophin is missing. And dystrophin plays a role in instructing that process. And that's really what Dr. Rudnicki discovered, is that that secondary job that dystrophin has is really related to making sure that muscle stem cells perform their function and produce new muscle tissue in response to damage. And so that really lines up with this idea of why muscle tissue is disappearing, right? It's not just getting damaged because dystrophin is missing in the muscle fibers, which is true. That is why the muscle is being damaged in the first place, but it's disappearing. And it's disappearing because these stem cells, they aren't doing a good enough job. And 3247 ensures that they can do the best job that they can.
Ryan, I just will add, because I know that I saw at least 3 or 4 questions. So this is a medicine that, as Ryan said, it's an oral medicine. You take it daily. And now what we're hoping is we actually don't think it's going to be every day. It potentially is going to be like 5 days of the week. Because if you think about like strenuous exercise or things like that, you need to take days off to let the muscle kind of naturally regenerate. And so our thinking right now is it'll be a daily drug where you take it continually over time, but it won't be every day.
Great, thank you. That was going to follow up with X. We had a few questions coming in from the chat about the frequency. And then if we go to slide 14, so you showed some of the data here from some of the limb muscles. We had some questions from some of the family members about potential for helping with the lungs, the respiratory function, and potential for any cardiac improvement. Are these things that you've looked at? Do you think this is something that SAT 3247 may benefit patients in those areas?
Those are great questions. So we have looked at the diaphragm. So the diaphragm is a skeletal muscle, and we don't have the images here in today's presentation, but we certainly have them in a poster that's available on our website that was recently presented at the World Muscle Society meeting in Prague. And you can see pictures just like this of the diaphragm muscle, where it appears that 3247 does have a very similar positive impact on improving the architecture or the structure of the skeletal muscle tissue in the diaphragm. We don't yet today preclinically have any data that speaks to pulmonary function, but this is certainly something that we're going to be looking at in the clinic in patients. It would stand to reason that improvements in the diaphragm could potentially have an impact on pulmonary function. We haven't yet looked directly for impacts on the histology of the heart, but what we can say, and Michael, maybe you can speak more to this if I miss something, is that we are fairly certain as stem cell biologists as a community that there is no true equivalent stem cell or satellite stem cell in the heart. So we don't believe the direct action of 3247 on stem cell biology will make an impact on any type of regenerative process in the heart. However, improving pulmonary function can be linked to improvements in cardiac function. And so again, we do believe there could potentially be benefits overall, but that remains to be seen in the clinic and is certainly something that we will keep a close eye on. as we move forward.
Yeah, and I also, and we have publications that address this in a different context, but we believe that improving mobility, improving muscle strength will, without question, improve circulation and make it easier for the heart to pump, put less stress on the heart, because movement and blood flow through muscles and the pumping action of muscles and movement on the blood system helps the venous return and makes the job of the heart easier and places less stress on the heart. So we think it could even have a positive effect on the heart, but not directly.
And then on the next slide, we had one or two questions just around the differences, and I know you touched on this, but sometimes helpful to reiterate, between the dogs and and again, as you clarified, the colored lines of the Satellos, the Sat 3247 dogs, different study than the dogs that are kind of used as the controls here. But the differences between the dogs in the two studies, and then maybe the rationale there would be helpful.
Sure. So, I mean, really, so what we've tried to do here is simply borrow information from the literature so that we could kind of benchmark the improvements that we saw. So it was very clear in our data set that we saw improvements in strength over time, from the beginning of dosing to 2 months, and then from 2 months to 4 months. But what we really wanted to know, because we only had access to the 2 dogs, was how can we think about the magnitude of these changes? And so in order to do that, you have to compare to something else. And so in this case, we looked at natural history data on the left, published by Joe Cornegie. Joe is very well known in the space for running the golden retriever muscular dystrophy model. His data is really unmatched in terms of quality. And so we thought that would be a great comparison to simply see how these force measurements we took stacked up against typical Duchenne muscular dystrophy dogs and healthy dogs. So that would be the rationale for performing the first study. And again, that's entirely separate and distinct from the study on the right, which answers a different question, which is, how does restoring regeneration compare to microdystrophin therapy, which I think is a very topical treatment in the landscape. People are very familiar with this therapy, and so we thought it would be interesting to benchmark against that data set. But no, there's no real comparison between the 2 studies. And I think there was a question regarding the different ages of the animals in the study on the right. And really, that wasn't by design. That's just simply the data that was available. So the dogs that were made available to us were 9 months of age. Typically, canine studies in Duchenne, the dogs are around 3 months of age when you first start with treatments, especially in the gene therapy space, because the idea behind the therapy is to preserve what muscle you have at that time. So in comparing a 9-month dog to a 3-month dog, we actually feel very good about that in consideration of the way the data came out, because this data suggests to us that the improvements which were seen from a 3-month dog receiving microdystrophin gene therapy were quite similar to the effect of treating a 9-month-old dog. So 3 times as old, a lot more progression, especially when you consider the natural history data on the left. Yet we see a similar, if not even more enhanced impact from 3247 on the older dogs. So, it's just a circumstance of this is what we had, so this is what we compared to. But I think given that we treated older dogs and had an equal, if not greater effect, I think that actually speaks volumes to how the potential of restoring regeneration. I certainly wouldn't see that as an experimental fault, if you will.
Thank you. I think that's really helpful, it's always a little bit tricky with, I think, preclinical data, and
Absolutely.
not something that everyone's used to seeing.
Yeah.
So if we go to slide 17, I think most of the rest of the questions probably can stay around here for them. So first of all, I think just to clarify, because I saw one of the questions in the chat, this first study that you're doing with the healthy volunteers, so that is not being done currently with individuals with Duchenne, but healthy volunteers. Can you talk a little bit about the goal of this study?
Correct. Yeah. So the first study is just being done called healthy volunteers. So these are generally adults who don't have a lot of medical conditions. And the goal is really to look at safety for kind of single doses as we go higher in the dose, as well as multiple doses. So over, say, 7 days to kind of see it's safe before we give it to, patients in the next step. And also to look at blood levels, right? Because we have a sense based on some of the animal data, how much level of the drug do you need to show a benefit? And so we kind of use both the safety we see as well as the blood levels of the drug to see, did it look as we predicted based on the animal work, to then guide us for when we go into patients to have a better sense. We can feel better about the drug we're using, the dose we're using, the dose of the drug we're using, and whatnot. And so that's the goal of kind of we call it a first in human study, where they get single doses and then they get multiple doses, again, to look at safety as well as blood levels of the drug. Because oftentimes what happens is you may kind of think a drug could last, you got to give it once a day based on your early work in animals. You then give it to healthy volunteers, and it turns out you got to give the drug 3 times a day to get the same amount. And so that's why that's the first step we do. And we felt it was important to first do this in healthy volunteers to look for these characteristics before we then move it into patients.
And then we had some questions around kind of maybe the safety or side effect profile. So either predictions from some of your preclinical animal studies or anything that you've seen within kind of this first healthy volunteer study in terms of maybe anticipated side effects.
Yeah, I think I saw a question about kind of almost what we call on-target side effects, right? The drug does act on a receptor. Are there known side effects? And so, our preclinical profile has looked very good. So far there's really nothing that we expect going in to see neurological or liver. Like we're not really expecting too much to be seen. Again, we've only dosed, a couple of our cohorts at the lowest doses. And so far we're very pleased with what we're seeing. The drug appears to be, generally safe and well tolerated, but this is also looking at kind of blinded data, right? So the early studies we have going on, there's still healthy volunteers, a certain amount get the drug and a certain amount get the placebo. So all the data we get to see before the study is done is blinded data. So we don't know kind of by the groups, but so far we're seeing very little, if anything. So it's encouraging, at least, as we're going higher in the dose patients or healthy volunteers for multiple doses.
And then there were similar questions about, is this muscle specific? Do you expect it? Is there any risk of kind of what we describe as off-target effects and other tissues that aren't the muscle?
Yeah, so we really don't expect too many off-target toxicities. Again, the good of it is it being specific to the muscle, and so far what we know from our kind of animal studies, we don't expect it. Obviously, that's why we do the studies first, and why we're doing healthy volunteers and going up in dose and increasing the duration to make sure we don't see anything.
And then as you're thinking towards kind of a trial in individuals with Duchenne, and it might be too early to have the decision of what exactly your patient population is like. We did have some questions around that, but also how are you thinking about SAT 3247 in terms of combination? Obviously we have standard of care tends to be patients on steroids, but we do have some families that have opted not to take steroids. If you've done any work with that or have any kind of expectation around how the drug would work with those.
Yeah, no, so those are obviously great, important questions, which I saw there were a number of questions. And so, a lot of it still will depend on what FDA wants us to do. And with drugs, the good of it right now is that there has been new drugs. And so I think that's the good. The challenging is us who is developing drugs. There are certain times where the FDA kind of mandates and says, we want you to just look at your drug without anything else. For something like Duchenne, obviously, we can't do that, right? So, we're going to allow patients to be on steroids, all different types of steroids. We're going to first look at people if they're not on steroids for whatever reason. And our goal, at least, again, we need to get approval from FDA, is to still look at it on top of kind of current treatments today, right? And so what we'll probably do, and again, we don't know the exact length or whatnot. I think people ask about maybe the ASOs or gene therapy. Even Givinostat was approved. Our goal is to test this in drugs that people are using, because we know people are going to be on these drugs. And so we probably are going to have a certain period of time for maybe a gene therapy. I think that's the hardest one, where maybe if it does take longer to show the benefit, we still want to be able to show that the benefit is from our drug and not because someone was maybe getting another therapy that had benefits. So we're probably going to have certain criteria. They have to be on a certain drug for a certain period of time to just kind of take out confounding factors. But our goal is to, test this on top of drugs that are available, because by the time we hopefully get to the market, there's going to be people on these other drugs. So, we imagine people are going to want to know if this drug can work on top of what they're already taking.
No, I think that's always helpful because it's a difference between is this from a scientific standpoint that you don't think it could work in combination, or it is from just a strictly trial design of how you're maybe first looking at it. And from
Yeah.
your view, it is not something that couldn't work with these other therapies. They could all be work in combination.
Correct. So mechanistically, there's reason to think it's, a different mechanism. It can work on top of other therapies. There's no reason to think it can't work with another therapy. So that's the good of it. And I know there's some other questions too about even other muscular dystrophies. And I think we do think there's the possibility that this drug can work in other things. We're obviously interested in exploring that. Our first focus obviously is in Duchenne. That's why we're here. That's how we're progressing. But the good news is we think this drug has potential in other muscular dystrophies as well. And we hope we get the opportunity to explore that as quickly as possible.
Yeah, I did see some questions come in around potential use in Becker, and then as well some of the other, like the limb girdles and some of the other kind of the broader muscular dystrophies. So could we just, I think you have it up here, but again, talk a little bit of, so we had some questions around, potential for participating in a study and how you were all thinking in terms of, we had some different questions around maybe countries and participation. So a little bit of maybe timeline that you're anticipating for thinking to getting to a trial in Duchenne, and, what your thoughts have been maybe around kind of design within, where would you maybe be looking, or anything you can kind of share in that realm.
Yeah. So our goal is, based on our timelines to file, an IND, which you kind of need to file to get it with the FDA to do studies in the U.S. specifically. And so our goal is to have 2 studies in 2025 in the U.S. The first one likely will be in older boys until we kind of shore up some of the preclinical work that you need to go younger and younger, which we're getting there, and then a little bit later on to go into younger cohorts. And as I said, we need to get approval and discuss this with FDA, but our goal is to do a couple different cohorts, go down to a low age range, potentially even 4 and up in one study, potentially 7 to 9 in another study, potentially 10 and older, and then do both ambulatory and non-ambulatory studies. So that is our goal for 2025. Again, assuming we get the blessing from the FDA, which we feel good about, is to look at kind of all different ranges of populations. And again, these will still be somewhat small studies to start, to kind of ensure that they're safe, to look at some biomarkers and obviously see if the drug's having an effect. And, we're definitely planning on being here in the US and currently running in Australia. I think we view it will likely be a global program, though. And I think there was questions about Europe. I think we know that we're going to need volunteers to go in these studies. So we anticipate, going into Europe. I think someone asked about Canada. Oftentimes, North America gets lumped into one study. And so I think, ultimately we're going to need to go globally to get the information we need. Again, we hope to study wide range of ages, both ambulatory and non-ambulatory patients. So we're going to likely need volunteers from all over the globe.
And I think partly too, I saw a few questions in the chat asking about, potential efficacy. And I think you already kind of alluded to this, Jordan, that part of this work is, that's what these studies are for, is to help us get to, you all learn a lot from your preclinical work in your animal models, and that helps inform what you're doing around dosing, around potential benefit. But, ultimately we need to get into the trials to really kind of understand the efficacy of the potential product.
Yeah, and I think as obviously everyone on this call knows, the FDA, I think, has been very open to new routes of approval and in moving things along well, which we're obviously excited about. We believe there still is an unmet medical need, and so these early studies are what we call learning studies, right? So it's to get the dose right, to ensure it's safe, to start looking for signs of improvement in muscle strength, motor function. I mean, a lot of the endpoints will be the ones, probably if you're familiar, done clinical trials, the time functional test, things like the North Star. Like we'll do those. Again, it's going to be still early to see if we see benefit. And then as we move to the later stages of development, I mean, again, we can't predict. We're hoping there still is potential for even accelerated approval pathways for additional drugs with biomarkers, right? We know MRI was very important to a recent approval from Givinostat based on publicly available information from the FDA's website. So I think we're optimistic that there's still potential quicker routes to get there. in the end, we're still going to have to show benefit on, an endpoint that's been used before, whether it be something like the North Star, or, now a lot of other companies are using time to rise velocity or forced stair climb, like Javinostat. So that'll ultimately be there. But we are hoping after we get this kind of phase 2 data that we do see some, interesting data, and we're hopeful that there still is potential, an earlier path to get on the market with accelerated approval. But, obviously time will tell what the landscape's like. The good news is there's a lot of drugs in development right now. And so, we don't know that the FDA's view is going to, be the same, say a year from now, 2 years from now, 3 years from now. But I think we're hopeful that there are quick paths for us to get this drug on the market if we're showing, potential signs of efficacy and the drug is safe.
That's great. And yeah, we've been encouraged and hope to see some continued flexibility from the agency as they evaluate products and see if we can get things to patients faster. I think that has been the bulk of the questions that were submitted during the talk and from before. If though you have additional questions that you think of after the fact, again, please reach out to us and we'll try to get you in contact and get those questions answered. And if you think this would be helpful for friends or family, we will have this posted up on PPMD's website in the next week. But I want to thank the Satellos team for joining. It was again, very informational and educational talk. Appreciate taking the time to be here with us, and look forward to hearing updates from you in the future.
Wonderful. Thank you very much.
Thank you very much, Eric. Thank you all.
Bye
Thank
now.
you.
Bye-bye. Bye, everybody.